Good afternoon, and welcome to today's webinar, Personalized Decision-Making in Breast Cancer, Why Prevention Matters More Than Ever. My name is Michelle Dixon, and on behalf of the Massachusetts Breast Cancer Coalition, I thank you for joining us today. I am very pleased to introduce today's speaker, Dr. John Urban. Dr. Urban is the Clinical Director and Associate Director for Clinical Science at the Tufts Medical Center Cancer Center and Professor of Medicine at Tufts University School of Medicine. He previously served as Director of Clinical Programs and Co-Director of the Gillette Center at Mass General Hospital, and was Associate Professor of Medicine at Harvard Medical School. Dr. Urban has published numerous articles, book chapters, and abstracts, and has served on several editorial boards, including the Journal of Clinical Oncology and the American Journal of Clinical Oncology. We are honored that he is joining us today. Dr. Urban, welcome.
Michelle, thank you very much for that very kind introduction, and I want to thank all of those who joined in today to hear what I think is an incredibly important and timely topic, that being how personalized decision-making has made our lives incredibly rich in terms of therapeutic options, but why it has emphasized how prevention matters much more than ever due to factors that we'll talk about. So in the next 20 minutes or so, what I would like to do is go through the problem globally and in the United States of breast cancer, what's happening in terms of our new therapies, which are very exciting, and think a little bit about how we may decrease the global burden and the local burden of this important health problem. So to start, as you can see, breast cancer in the United States is an incredibly important and common problem, and cancer in general is. For both sexes, over 1.6 million estimated cases of cancer in the United States. 500,000 deaths have been attributed to cancer in the United States, or estimated that this will occur this year. And as you can see, in breast cancer, approximately 250,000 people will be diagnosed, some men, but mostly women, and approximately 40 to 41,000 deaths will be attributed to breast cancer this year. The problem, therefore, is an incredibly important public health issue that we all have to think about in terms of both better treatments and prevention. If we break it down a little differently by age, decade, you can see that invasive breast cancer is a disease more commonly seen in the older population, but it is clearly a public health problem even in very young women in their 30s, where issues of family, issues of childbearing, issues of best treatment, premature menopause becomes a very important health issue. 10,000 invasive cancers per year in the aged less than 40. And as you can see as well, pre-invasive disease known as in situ cases is not as common, but is still an important issue, because we want to not only treat aggressively where necessary, but we don't want to treat where it's not necessary, and in situ cancers make up a fairly important percentage of the burden of disease. Looking at these data a little differently, you can see that while breast cancer is the most common cancer in women, lung cancer remains the most lethal cancer, but close behind is breast cancer, accounting for approximately 14% of cancer deaths in women in the United States. Now, at the very beginning of the talk, there was the lead slide. I had the picture of an elephant, which has many meanings in this talk. Jumbo was the mascot for Tufts University. But the reason that the elephant appears again is that it is not inevitable that a mammal on the planet Earth will develop cancer. But I did want to show some comparative statistics. If you look at men up until age 49, 1 in 29 men will have developed cancer. And if you go to age 70 or so, 1 in 3, and from birth to death, the lifetime risk of developing cancer is 1 in 2.5. For women, 1 in 19 by age 49, and 1 in 3 women lifetime will have developed cancer, which is an incredibly frequent problem. However, when you look at the elephant, our friend the elephant, only 1 in 20 elephants lifetime may be diagnosed with a condition in the family of diseases known as cancer. And so it raises lots of questions because elephants live a long time. They have their offspring late in life. They clearly have 100 times more DNA than we have, and cancer is a disease of DNA going wrong. And so the question is, why, with so many things that could go wrong, why is it that the elephant doesn't? And there are many biological reasons why this may be the case, one of which is they perhaps have many more copies of the tumor suppressor gene known as p53, but it does raise some other hypotheses as well. And if you look at women breast cancer, just to show some comparative statistics, 1 in 53 women up to age 49 will have developed breast cancer, approximately 2%. And on lifetime, the statistic is approximately 1 in 8. So this is, again, a very common public health issue, and we need to know more and do more to prevent it. Now, globally, and in China, as an example, this is a huge public health issue. Cancer is predicted to occur in over 4 million women, 4 million people in China in 2015. Approximately 12,000 new cancer diagnoses every single day. The most common cancers are shown in the next bullet. And in women, breast cancer is among the most common diagnosed cancers. 15% of all new cancers in women are breast cancer in China. And the mortality statistic is really stunning. In 2015, it was estimated that almost 3 million Chinese would die from cancer, counting up 7,500 cancer deaths on average per day, with the leading causes of death shown below. The 5-year survival of cancer patients was estimated to be about 36 to 37%. Women having a slightly higher likelihood of surviving cancer than men. And in terms of the global burden or the burden in China, it was estimated that perhaps 60% of deaths from cancer might be avoided by trying to limit or avoid modifiable risk factors. And those factors that I've shown on this slide include chronic infections. We know that hepatitis B, hepatitis C, HPV virus, H. pylori, which has been associated with ulcers and gastritis. These may account for as many as 3 out of 10 cases of new cancers in a country like China. Smoking might be responsible for as many as a quarter of all deaths, which is easily a modifiable risk factor. And there are now other advances that have suggested that diet, exercise, and environmental exposures may account for a substantial fraction of disease. So when we're thinking about reducing costs and improving the quality of life, reducing the death rate from cancer, and helping the whole health system avoid the shock of the expense of treatments, it seems very logical to focus on prevention, and prevention would include many of the factors shown on this slide, and all of which are somewhat under our control. So if we show this slide in a little different fashion, we have the human genome, which is important base knowledge of our DNA. We have modifiable intrinsic risk factors, what I'll call intrinsic, meaning our own body, that we can focus on diet, on the obesity epidemic, exercise, and the smoking. And then we have so-called environmental exposures, such as exposure to chemicals, toxic air, and we have really a lot of questions that still need to be answered here. For example, when is the exposure important? Is it important around the years that you develop the cancer, or is it important 20 years earlier, or even before we are born? There are clearly experiments that have suggested that a brief exposure of the fetus to a drug or a chemical in utero can affect that child's or adult's risk of cancer years or even decades later. And so these kinds of experiments make it clear how difficult it is for us to really equate an exposure to an outcome and how many years it takes to know what is going to happen. So as a sign of what's happening, this photograph I took actually on a trip to Shanghai about 2 years ago, a little over 2 years ago, and what it really shows is the air over a beautiful area of the city. It looked a little hazy, a little foggy, and it was a perfectly sunny day, except that there was really a suggestion of significant pollution in the air during the week we were there. And the World Health Organization said that recently that 92% of people breathe what is classified as unhealthy air. And that, and most of those people, nearly 2/3, live in areas of Southeast Asia, Western Pacific, less so in Europe, and this problem may account for as many as 3 million deaths a year related to a number of different health problems. So obviously, if we can see the problem, we can expect that there is going to be major health outcomes, but there are many times where we may not be able to visualize or see exposures that are probably going to be increasingly important as we learn more. So what kind of evidence is there that exposures matter? Well, we have these two very simple experiments. One is the old DES experiment, where in utero exposure that's brief or months by the mother taking DES to prevent early delivery may cause cancer 8 to 40 years later. A rare type called adenocarcinoma, clear cell adenocarcinoma, was identified in women, and clearly a rare disease that became a slightly more common, was the signal for thinking that this exposure could be a true problem. And there's been some debate that breast cancer risk may also be affected in women over the age of 40 to 50, suggesting that an exposure 50 years earlier might change one's risk of breast cancer, a very low-level chemical that might affect one's health years later. Recently, very recently, this publication just came out. Animal models have suggested that if you expose an animal in utero to certain types of estrogen-like compounds, it may induce a resistance to a drug that we use to treat cancer if cancer develops through some epigenetic changes of the genes. So this is a suggestion that these exposures might not necessarily cause cancer, but that they may cause the cancer to be more resistant to treatments that we currently give. If we look in the pre-precision medicine era, this precision medicine I would define as taking patients' tumors, sequencing molecularly, see what they have. If we look in the era before that, you can see that happily mortality from cancer was falling in certain types of cancer. So we can show that all of the great excitement in precision medicine, that we can do things and have done things to lower the rates of death from breast cancer and other cancers by doing things like focusing on prevention, like focusing on nutrition, health, and other types of therapies that have been very, very successful to this point. Looking a little bit differently in terms of breast cancer, you can see that somewhere starting around 1990, the mortality has decreased quite substantially, happily, and that we're seeing certain other types of cancers, like colorectal cancer, where the drop in the decline has been really steady and important. So it's not all about precision medicine that is going to help improve the outcomes. It's about many factors and many ways to get to the goal we all want. These are some of the drugs, therapies that are currently in use for breast cancer. You can see that surgery and radiation have been still very important. Many other types of therapies that are non-drug, but within the drug area, we're seeing hormone therapies, chemotherapies, biological therapies, these very exciting immune checkpoint inhibitors, the HER2 type of drugs that have really helped patients with HER2-positive breast cancer. New drugs such as PARP inhibitors for patients who have the BRCA1 or 2 type of breast cancer, and there are certainly others. And this has taken what used to be a very uniform treatment, one size fits all, and broken it down to many, many exciting and yet costly types of agents. And this slide just shows one thing, which is not to get sort of too bogged down in the details, but if you take a single test, now in this slide basically shows on this line the cost of the test here, and as the cost of the test goes up, the cost per year of life saved of using the test changes significantly. And if you talk about a woman who's 30, 40, 50, 60, or 70, applying these tests can have a meaningful difference in cost. This test, let's say at $4,000, might cost $27,000 per se for a year of life saved by using it if you were in your 30s. But if you were in your 70s when it was applied, it could cost over $175,000 per year. So these tests, these therapies all have great value, but they also have significant costs associated with them depending on how they're used and how often. And we know that for certain therapies that seem very simple, such as taking a medicine known as letrozole for an extended period of time, we may reduce the incidence of recurrences of breast cancer and decrease second primary breast cancer is a very important endpoint. But in order to do that, it may, for example, cost over $200,000 per year of life that we save, raising the question, do we really want to be treating more and more cancer successfully, or would it be much more cost effective to think about prevention and avoiding the disease in the first place? And those are sort of the questions that these kinds of cost analyses have raised. Again, lots of different drugs in the chemotherapy world have become increasingly effective and increasingly well tolerated, so we've had a lot of successes there. But in the personalized therapy approach, you can see this diagram, which essentially shows that loads of places exist in a cancer cell to try to interfere with a cell growth or cell division. And by doing so, if you can find a weak link, you can have tremendous success in reducing the growth of the cell, in reducing the tumor burden, probably not necessarily eradicating all evidence of the disease in most cases. But examples up here where people have used the HER2 protein and a drug called trastuzumab, also known as Herceptin, and the Herceptin-like drugs has shown great success in reducing the burden of disease once it's developed and improving the success rate of treatment. Those of us who remember long enough back will know the first targeted therapy, the very first personalized therapy, and that one of the most successful of all time would be a drug called tamoxifen, which is useful for ER-positive, hormone receptor-positive breast cancer, and is now a prevention drug. But it does no activity at all if the cancer is not sensitive to estrogen. So that is in the form of a targeted therapy. But all of these different places where you can intercede lead to places where a drug can be developed and maybe help reduce the growth of the cell at significant personalized cost. So personalized medicine assumes that individual therapy will be directed by one or more mutations or changes. The driver mutations can be identified, that they're present in all of the disease, so that everywhere there might be some disease in a patient, that it all would react well to the drug, that the mutations are stable. We wouldn't want to start a drug in 3 or 4 months, find that the mutations change, and all of these things are problems with the approach that I'm just identifying. Driver mutations have actionable drugs and therapies. We have knowledge of some of them, but there are many more to discover. Actionable mutations will predict behavior across diseases, meaning if it works in breast cancer, would it also work in colon cancer or lung cancer? And that to date has not turned out to be the case. And that we really want is that drugs will have low toxicity, and that includes toxicity of the side effects, but also financial toxicity. Now, I show this slide, which was another, on the same trip to China. We visited Taiwan, and we were in Taipei, and on one day, all of a sudden, in this block, all of these pandas appeared to celebrate the birth of a new panda. And the point is that in years past, we would treat all of our population of women and men with breast cancer with virtually the same types of treatments, one size fit all. And doing so, the pros of that were it was uniformly delivered, and it was variably effective from one person to another, but the cost could also be scaled and was moderated by that. But the downside was that it was not effective for nearly as many people as we would like. And so we've gone from this approach of treating one size fits all to treating everyone, every individual separately or personalized by their own approach. And the cost of doing this is substantial. Even 10 years ago, the New York Times published that each of these drugs had a substantial personal monthly cost, $9,000 a month for, say, Erbitux, $7,000 for a drug called Avastin, which is no longer commonly used, but substantial. And if you look at a more careful analysis of this, that in the year of approval, a 1-month cost of a new cancer drug was approximately $1,000 per month. By 2015, that cost became approximately $12,000 a month. And by comparison, in normalized dollars, the average annual U.S. household income was approximately $48,000 in 1984, and 30 years later is $53,000, barely a change, clearly not 12 times as much. And so we really all have to struggle to make sure that we understand that treating our way out of this is not only difficult from the point of view of making an impact in real time quickly, it's also going to be extremely costly and is hard to scale when we really decide that every individual is going to need a personalized approach. So the cost of progress is shown really on this slide. We're now at $10,000 to $12,000 per month for new cancer drugs. Oral drugs use is growing rapidly with substantial co-payments and out-of-pocket expenses. Many drugs come in a one-size vial, and so these drugs are just unfortunately wasted when not all of them can be used for the right individual, and that accounts for $3 billion a year. And bankruptcy is really associated with worse outcomes and increased mortality, and so we have to be thinking a lot about this burden. The NCI and others are really trying to do better. We're trying as scientists to do a lot better to try to match the therapy to the patient, which is really the heart of personalized medicine. And it is difficult, but it's very promising. By taking a person's tumor and sequencing it, finding the weak links, we're looking to find single drugs or combination of drugs that might be highly effective for individuals. But as I've just shown, this may be a very promising medical approach, and we all are excited about the potential. We have to think about how to make it scalable for every individual. A second study that's being done is trying to assess whether or not this approach is really going to be the best way to go, in which case the tumor is sequenced, the mutation is detected. Half of the time the patient will be treated with a drug targeting that mutation. The other half of the time the patient will be treated by the best clinical judgment that the oncologist has, and we're going to see whether or not one side is more effective than the other, or vice versa. That this is a promising study that will really help us to know how often our judgments are the correct ones with or without this sort of sequencing type of data. So what's happening though in I think our field is that even most recently this week there was the Moonshot Initiative that was so strongly being proposed by Vice President Biden and others. Really fantastic effort to try to focus our efforts on collaboration and integration of resources and talking to each other. Still, I think we have to think that among the priorities has to be research on the cancers, but also has to be research on the prevention of the cancers. And even back years ago, there have been many speaking out, talking a little bit about the problem of prevention, and we have many different factors in the environment that can be focused on, such as our own behaviors, smoking, alcohol intake, but also exposure to chemicals, pollutants, products that we eat, or package our food in, drugs exposures, radiation, socioeconomic factors. Many of these factors will, with the proper study, be associated with risks of cancer at a low level, admittedly, but across a broad population, small changes in exposures at critical times in one's life may in fact have a major effect on the burden of cancer going forward. And again, to show what the environment can be like in some parts of the world on an average day, this type of exposure is difficult to perhaps change, but is so important because of the prevalence of disease and the global burden that we are all facing and treating. So what's the final message, I guess, on this slide? And I really applaud Silent Spring and other institutes like it that are really focusing on hard, non-debatable, rigorous science. We would really need more data, because what we're all asking for is, what can I do that is not just something we can do, but something that can truly impact our outcomes? What can I do to help myself, my family, and others? So I think the message here is that we are seeing great progress in cancer in terms of mortality, and our therapies are better, less toxic, more effective. And cancer in all mammals is not an inevitability. We don't have to experience this. The major work in our understanding of pathways is ongoing. As we get more and more smart about individualized cancer therapy, we may have great successes, and we're seeing that, but we may also have the successes come at significant cost, and in some cases significant side effects. Early detection, how important it is still not prevention of the disease. It's just simply finding it at an earlier point where the treatments may be more successful. But prevention is going to require much more understanding of our exposures, and we're excited to hear Silent Spring beginning a trial, a study really of personalized environmental exposures, personalized testing, to see whether or not we are exposed, and once we know the burden of exposure, to think about how we can change that in cases where changing the exposure could potentially have outcomes. I'm cautioning that we shouldn't immediately assume that changing any of our exposures will make a difference until we really can equate and relate these exposures to health outcomes, but that's going to come from good science as long as it's funded well and we focus on it. And personalized medicine will require more than just matching drugs to mutations if we are able to afford care for everybody going forward. So I thank you for your attention, and I'd be happy if there are questions to address them. Thank you very much.
Thank you, Dr. Urban. I would invite anyone who has a question to please type it in, and we can share them here. I have got one that's come through. Can you just discuss a little bit what the practical ways are of reducing one's risk of breast cancer?
So,
specifically as it relates to reducing one's exposure to toxic chemicals of concern.
So thank you for that question. It's really an important one. Just briefly, I would say that important factors, really important, and it's not just feel-good advice, but truly important factors that help reduce risks of breast cancer include maintaining vigorous exercise. Exercise biologically in the body has very potent anti-cancer effects, so moving is important. Caloric intake, being careful about diet intake, and avoiding, of course, obesity, whatever the caloric intake. In terms of exposures, we're surrounded by ways in which we can have exposures, some of which are low level, others are more sort of obvious, as we saw in the example of the air pollution in China. But simple things that could be done. We know from experiments that Silent Spring has done that drinking certain types of liquids out of certain types of containers, by just changing the types of containers, one can lower the level of exposure of certain compounds in the body in healthy students in a matter of days. What that means in terms of health outcomes is not clear, but I would say that if one were trying to think about practical things, one might, for example, avoid microwaving in plastic containers where high temperatures and contact with food and plastic containers might lead to some leakage or some chemicals at a low level that could be in food, as an example. One can certainly be cautious about the nature, the type of water that they are drinking, what types of containers are bought to carry it, to store it. One could also, of course, avoid any kind of exposure to cigarette smoke or other environmental pollutants that can be identified. So there are some common sense things. I would really be cautious about taking the evidence further. Do we know at this point that any of those will make me a healthier person? No, but I think the precautionary principle applies, which is until we know more, we should assume that we want to be a little cautious about the kinds of exposures that we can minimize. There now has recently been in Boston a regulation that removes flame retardants from furnishings. Flame retardants used to be commonly added to furniture to avoid fire and to reduce the risk of fire, they've been shown to be released in fires and to be highly toxic. By removing certain chemicals from the manufacturing of certain types of products, it will be interesting to see whether or not we can, over time, make the environment safer in that regard.
And along that lines, I have a follow-up question that comes in. How much do the question of toxic chemical and their role in the development of cancer get raised in research meetings like AARC and ASCO?
Yeah, that's a great question, and it's really an important one. I will say, even in this week's announcement, if we looked at 10 major points of potential progress for the moonshot, I would say only, to my recollection, one of them really addressed the aspects of prevention science. So at the meetings, happily, until recently, it was not very common that you would go to the scientific sessions and focus much on prevention science. But happily, over the last 2 or 3 years, we have seen a great deal of interest in some of the institutes in the whole field of prevention. And I think that it has been largely spearheaded by the realization that the costs that are being experienced both to the health system and to individuals are really going to become prohibitive, and that the clear way out is to really focus on maybe investing in the science of prevention. It's a very hard science because obviously the outcomes sometimes have to be measured in days, years after the exposure, let's say, and prevention is really the absence of something happening. So it's hard to do that science, but more and more and more you're seeing enthusiastic attendance at sessions in the major meetings focusing on prevention. I think where it's taken a lot, where it's much further ahead at this point is in the relationship, for example, of diet, exercise, obesity to the cancer epidemic, and in some of the infections that have been associated with cancers. I think we're going to see a lot more excitement in personalized testing to exposures, and maybe certain exposures that may influence rates of cancer in the next many years.
Great, thank you. If there are any other questions, please feel free to submit them now. Dr. Urban, can you speak a little bit on the status of personalized exposure testing?
I think that this is a very exciting area, but we have to be cautious not to over-interpret whatever the data are. I think one of the best projects ongoing is a project that is just now starting through Silent Spring, and others are thinking about this as well, where testing for certain compounds that have been shown to have biological effects. There's a whole class of compounds known as estrogen endocrine disruptors, where the signaling of a hormone in the body, let's say estrogen or androgen or some other hormone that can cause developmental changes in cells or be associated with events like cancer. For example, the use of estrogen, postmenopausal women with estrogen exposure may have a slightly higher rate of breast cancer. These exposures, these chemicals can have certain effects, much like our normal hormones. And so Silent Spring and others are now just starting to do individualized testing through a novel type of protocol. The results will be helpful to understand what the prevalence of exposures is. They will not necessarily be able to guide a person as to what they should do to lower their exposure or what should they do to improve their health. But if there were certain compounds identified that have biological effects, a person might be able to make the decision that they might change certain habits or behaviors to see if that compound or exposure changed, and if it did change, how, what could they do to follow their the outcomes. So personalized testing, I think, is really the next idea in personalized medicine. We know our DNA. We know how the DNA may change due to the disease, and we know that the DNA in our bodies is interacting with other factors that are exogenous factors, and we just have to understand more how these compounds and chemicals and dietary changes and exercise changes affect the behavior of our DNA.
Okay, thank you. See if we have any more questions. Well, Dr. Urban, I thank you really on behalf of our Executive Director, Cheryl Osimo, and our Board of Directors. Thank you to both you and to Tufts Medical Cancer Center really for all of the ongoing support that you have shown the Massachusetts Breast Cancer Coalition in supporting our educational program, Let's Talk Prevention, and obviously the work that you're doing with our sister organization, Silent Spring Institute. We thank you for joining us today. This has been incredibly interesting and insightful, and we appreciate your time.
Michelle, thank you very much. Thanks to the Mass Breast Cancer Coalition for all of the incredible work they have done over the many decades, and I think that the progress is fantastic, and I think we're going to see some very exciting work in the next 10 to 15 years, and your support of the effort has been really a major reason why. So thank you very much.
Thank you. And thank you to all of you who have joined us today, and have a good afternoon.
Thank you.